next step is the process of translation. We have started out with DNA, and through the process of transcription that you just learned about, we manufactured messenger RNA, which we then, as eukaryotes, modified by adding a 5' cap, a 3' poly A tail, and by removing the introns. And now we're going to see what happens in the next step, translation. This is where I actually read the directions or codons in that messenger RNA and use them to direct the manufacture of a polypeptide chain, which will eventually become a protein. So translation is a process by which we translate those triplets of nucleotides into a sequence of amino acids. And there are a number of different players involved in this process that we're going to need help from in order to get to the end of our goal here, which is the making of a polypeptide chain. We're going to need to have transfer RNA involved. This is a molecule here of transfer RNA. You'll notice it kind of has a T shape to it. It is a molecule of RNA that has been folded into this shape, and it has two regions to it that are of importance to our discussion. On one end of this tRNA, there is a location where we can attach an amino acid. Amino acid. On the other end is a region of RNA that is a complementary sequence or triplet that is complementary to the messenger RNA, and each different tRNA has a different complementary region. This complementary region is called the anticodon because it's the antithesis or the complementary base pair to a codon. In this case, this particular tRNA is the anticodon GAA, which means it's complementary to the sequence CUU on the messenger RNA. So that's one player that we're going to be discussing. Another player we have to have, obviously, we have to have that mature messenger RNA after it is exited out of the nucleus. And then the last thing we need to have is something called a ribosome, which you've learned about already. Remember again, ribosomes are made up of a little bit of ribosomal RNA and a little bit of protein. The purpose of the ribosomes is simply to set the reading frame. It's to basically set up how the nucleotides are read in groups of 3 so that we read the correct groups of 3 so that tRNAs can match up to those groups of 3 with their anticodons and bring with them their corresponding amino acid. So let's see how this all works out. Now, to start the process of translation, the first thing that we got to do is we've got to have our messenger RNA and we've got to have a ribosome. Now, you didn't know this before because we really didn't mention it too much, but all ribosomes contain 2 subunits. And those 2 subunits, when a ribosome is inactive, will be floating around separate from each other in the cytosol of the cell. And it's only when those 2 subunits join together that I get a functional ribosome. In eukaryotes, the 2 subunits are called a 60S and a 40S subunit. The 60S subunit is the larger of the 2, so it's called the large subunit, and the 40S subunit is the smaller one. So for example, here would be the 60S subunit, here would be the 40S subunit. In prokaryotes, we also have a large and a small subunit, but they weigh just a little differently, and they are 50S and 30S. Okay. Now, when I am getting ready for messenger RNA to be translated into a polypeptide chain, I'm going to have to be out in the cytosol. I just want to remind everybody of that again. And that is the location where you find all these ribosomal subunits. So let's look and see what happens when messenger RNA exits out of the nucleus and comes into contact with a large and a small ribosomal subunit. So here we are. Here was the nucleus over here. We already went through the process of transcription in the nucleus. We've made our messenger RNA mature. It's exited now out through the nuclear pore. You can see right there. And now you'll see that a large and a small ribosomal subunit are beginning to attach to that messenger RNA at a special location I'll get to in a second. And then we're going to see that ribosomal RNA come come together, the two subunits, to make a functional ribosomal unit, and that ribosomal unit is then going to serve as the reading frame for bringing in transfer RNA. So let's take a look at this. We're going to go through 3 steps. They're going to sound familiar to you. There's going to be an initiation phase, there's going to be an elongation phase, and there's going to be a termination phase. So let's start out with initiation. In the initiation phase, here's my messenger RNA in purple. The first thing that happens is when that mature messenger RNA gets out into the cytosol of the cell, a small ribosomal subunit will attach to it near the start codon. The start codon is the first codon in the polypeptide chain, and the start codon, you need to know, is always AUG, and that always codes for an amino acid that brings, a transfer RNA that brings in the amino acid methionine. So AUG codes for the amino acid methionine, and you can see here our small subunit has attached. You can see that immediately following the small ribosomal subunit attaching, a transfer RNA with the complementary anticodon to AUG has attached, and it brought with it this methionine amino acid. Now, next thing that happens, we still don't have a functional ribosome yet because we're missing the large subunit. The large subunit will now attach itself over top of this transfer RNA and attach itself to the small subunit. So now we have a functional ribosome here, and in that functional ribosome there are 3 regions called an E site, a P site, and an A site. Those are going to be it's critical for us to knowing what's going on here in the next slide, but there are 3 sites, and when we start out here, the methionine is in the P site. So let's look at what happens next. That's the end of initiation, by the way. Initiation is just getting the small ribosomal subunit and the large subunit attached and getting that first transfer RNA into place. Now, during elongation, a second transfer RNA molecule is going to show up that has the complementary anticodon to the next 3 nucleotides in the sequence of this RNA. In this case, I think it's a GAC, it looks like there. So we're going to have a transfer RNA come in that's complementary to that in its anticodon, and notice it brings with it a particular amino acid. Okay, so that new tRNA that comes in always comes into the A site. That's the first place a tRNA goes once we get into elongation. You can think of A as the add site. This is where we add a new transfer RNA. Now, the next thing that's going to happen is, here's my methionine as well as some other amino acids that have been attached that are in the P site. Those are all going to, once we get this transfer RNA in here, we're going to move our growing polypeptide chain from the P site over to the A site. So here you see, here's my growing polypeptide chain. We're going to transfer it over and stick it onto that new amino acid. So we're going to transfer it from the P site into the A site. Now we've got a big old long chain in the A site, and now the whole ribosomal subunit is going to shift forward. And what that's going to do is it's going to take this guy here that was in the P site, and it's going to put it into the E site for exit. You can think of E site as in exit. Okay, so there's that. This guy's going to go into the E site, and the long chain here that was in the A site, you can see has now been shifted into the P site. And now comes in the next amino acid that's the next transfer RNA with its amino acid that has a complementary anticodon, and it goes into the A site where we add a new transfer RNA. The next thing that will happen is this long chain that's in the P site will get transferred to the amino acid in the A site. The ribosome will shift down again. The last amino acid tRNA will get booted out at the exit site. We add the next one in at the A site, and we continue this process of elongation until we reach a point where the ribosome encounters one of 3 different codes these are called the stop codons. If it reaches any of these three sequences, something different is going to happen. And let me just show you right up here, if I go back here for a second, on that table that we looked at, back just a little more here. Yes, there it is. You'll see on this genetic code table that there were three sequences here that did not code for amino acids. They all coded for the stopping of elongation, and those were UAA, UAG, and UGA. Those are called the stop codons, and we are going to be relying on those to tell the ribosome when it's time to stop elongating and to terminate the manufacture of this protein. So here it's reached one of the three stop codons, and what happens is, rather than having another transfer RNA come in, a different kind of protein called a release factor comes in, and the purpose of this release factor is simply to dissociate the large and small subunits from each other so that they release the messenger RNA, and so that they also release the polypeptide chain that's been manufactured. And that's the end of the manufacture of the polypeptide chain. Now, one quick point, it's not a mature protein yet. Other events have to happen to that protein to make it mature. It has to do with secondary structure folding. That's the making of the alpha helices, the beta pleats. It has to do with tertiary folding patterns, and it usually has something called chaperone proteins that help it do that before it's finally a mature protein. But in terms of the central dogma, we have covered everything you needed to know on transcription and translation. Next up, we're going to talk about what happens if we make a mistake during transcription or during DNA synthesis and a mutation arises in the DNA or RNA.